This is our first national simulation. Simulation is something you will keep repeating to make sure that by the time anything happens, you move like, as I say, you move like the people on ER, in the emergency room. So morning everybody, welcome to Bodles. Um, we are about to go inside, but you have to observe some protocols, so we just need to make sure that everybody hears and understands. Stand in the shade. Um, Let me just tell you how we're going to proceed. Agenda by words. We're going to start with a prayer. Then I'm going to explain to you what today is about, give you some instructions. And then Mr. Lewis, who is in charge of farm biosecurity, will explain the procedures from you enter. And then when we get inside, Miss Henry Myers will give us a little talk on what the disease looks like to fresh up your mind so that when you watch the procedures, you understand. And we did all of this from countless videos of simulations by others. Debbie went to Belize, looked at simulation. We are in the process with PQPI, fine-tuning our PRA, but we looked at every risk. So the simulation really is just to go through what we do the minute we have a diagnosis of positive. There are many more aspects to the simulation that we're not doing today. For example, we need to look at the borders, and we're going to do that simulation very soon. But we don't have to do that today. We also will look at the simulation also looks at our laboratory procedures for surveillance and testing. We're not doing that today because obviously that is in Kingston. But today is a crucial time. The crucial simulation is to look at the field after we get a diagnosis, what do we do in the field? So it's just to get that. We want to explain the process so you understand, so that if you hear we're doing a simulation, it does not necessarily mean the disease is here. Because when you go inside, you will hear that we do actual eradication of the disease once we get a positive on the conventional PCR, a positive on the real-time PCR, and everything is repeated, and a positive on what is the confirmatory test here, which is the LAMP diagnostics. We cannot wait for the confirmation overseas. It takes a while to go and to come back. So if we have those three confirmations, we'll follow through with the process that you'll see in there. So I'm just telling you this because a lot of the things that you hear about in other countries that, oh, this place has, it's because this is what happens. And then by the time the confirmation and reconfirmation overseas comes back, it was a false positive. But we do not have the luxury of time to wait on that. Usually the area that we actually take out, and we are trying to tell the farmers this is something that they must expect. This is how we safeguard what we have. So I'm just telling you that. I also want to reiterate for everyone, the disease does not affect the fruit. So even if a tree came down with the disease and it had a fruit on it, it will not go up in the fruit. The fruit can be eaten. If you have a field that is infected and the farmer has mature fruits in it, it can be eaten. So be mindful of that, and I want everybody to understand that. This is a disease that affects the crop, but in affecting the crop, it can wipe out everything. So before I go further to give you a fulsome overview, I just want you to understand that. It's a fine-tuning of the simulation process. Your feedback is important. At the end of it, you're going to get a sheet, and you can tell us too. You know, if you feel it's too long to write to explain how badly the thing is, just let us know. We don't feel bad. This is our first national attempt. The next time we do it, we might invite everybody. We will try and perfect it. So this is the reason for inviting all of you. You are our feedback. Just to say that when we go, Mr. Lewis is going to explain what you need to do for farm biosecurity. It starts at the gate. So he'll tell you what to do. However, just want to prepare you all that when you go in, and we'll have a little time to put on hazmat suits if you're going into the area that we're going to show you how we do the eradication. If you're going to put on the suits, you cannot have your bags and stuff with you. So if you want to leave them in your vehicles or you want to take them with you,
Office.
we didn't actually put a space for bags, but you can put them in somebody's car. One vehicle is going to drive in, two vehicles going to drive in, because we really don't allow a lot of big vehicles to go in. So just bear that in mind. You can decide how you. I know my personal is my personal, so everybody's personal is their personal. So you're gonna go in, and we're gonna put on hazmat suit. So just to tell you that. Just want to tell you that this is just simulation is one of the components in our national plan. It has other areas. We have border security, we have simulation, we have farm biosecurity, we have surveillance and diagnostics, we have the PRA, and we have other areas that we need to beef up.
Communication.
Public-private partnership, how we're going to deal with this disease on a sustainable basis, should it come. And if you're listening on the radio, you hear a lot of PR on the radio. We're going to try and hold it again. We're doing it now. We're trying to get a hold some for when everything opens up in a wider scale. People are traveling. I'm sure you all heard. It's now in Peru. It's moved from Colombia to Peru. There was a scare in Venezuela, but Venezuela is actually negative. And everybody who joined the webinar on Tuesday was bombarded by, or Wednesday was bombarded by this Zoom bomb. So they're going to do it again. And that really was giving us a whole perspective on the status, and especially looking at Venezuela's preparedness, which I was quite enjoying. Venezuela is preparing theirs with Bolivia, which makes sense because they share borders. So we are heightened here because Colombia, as you know, people move from Colombia to here, formally and informally. We are collecting samples, we are sending them overseas. Just to know that Sheree will speak when she gets inside and she'll talk about the diagnostic side. We sent out some information to you about a paper by Villahen. Villahen is actually the person who is reconfirming for us. And he wrote a document on diagnostics so far for Mozambique, which is where he came. So we have our systems in place should anybody know of anything. And we get a lot of calls for all kinds of things, which is not it yet, but we welcome them because we send somebody and we collect the samples and we follow through. And we do send samples away. So, but even if, let's say, let me just tell you, we are extremely cautious. We're not going to send it away only if we get 3 positives from the 3 tests. If we get 2 and 1 is different, we send it away to make sure we get. So we are doing that. So when it's positive, it's positive. What we don't want, since you're all here, and we want a consensus on this, we don't want the rumor mills to go around before the thing is here. We want you to follow the process. And the process is, when we know, we will follow the protocol to inform our counterparts and our principals. So when you hear it, you will know for sure. So don't listen to any rumors. And if you hear a rumor, call us. It's like you're going to call us if you think you see this, call us. And we will put it to rest. The worst thing you can do for an industry is to kill it with something that is not there. Okay, so just bear that in mind.
Thank you all again for being here. The issue of farm biosecurity is a very vital area in the fight against TR4. And today we have 4 biosecurity control points that we'll focus on. One, we have the quarantine area, which is at the gate, which is very symbolic because this is one of the key areas in which you can have the TR4 enter farm or leave the farm, okay? So the other areas, we're going to talk more about them when we get in, but those areas are relative to the infected plant that we have simulated. So they're zone A, B, and C, and there are different quarantine checkpoints there, biosecurity points. But for this area, one critical area is the visitor register. And I guess I trust that most persons, everyone here, have been registered. But that is very important in farm biosecurity, and we're encouraging all farmers to do likewise, because this is a very important tool as far as traceability is concerned. All right, so what is going to happen today is that we're going to allow just two vehicles to enter the premises. All of the vehicles will be here, and we will, at some point, we'll observe what is happening. We're going to ask all persons that are here to utilize a footbath. So the footbath and the vehicle bath, there's a vehicle bath at the entrance, and there's a footbath there for those that are on foot. Both the vehicle bath and the footbath, there are two products, quaternary ammonium compound and glutaraldehyde. These are one of the best treatment disinfectant for TR4. And so those are in the footbath. Whenever you're using a footbath, I want to emphasize that you should spend 3 seconds, not 3 minutes, 3 seconds. So we say 1, 1,000, 2, 1,000, 3, 1,000. 3 seconds in the footbath. And likewise, when we head inside, the same principle will apply. We'll talk more about that. The principle here that we have to ensure is that we want to go inside clean. That's clean of TR4. And when we're exiting, we exit clean of TR4. And so we'll use all of the protocols to ensure that takes place. When we are through here observing what is taking place, so you're gonna see the power wash that we use to disinfect the tires, because you want to ensure that you remove as much soil material to ensure that the disinfectant works. It works best with less soil. And so the power wash will be utilized, and the tools will be immersed in the disinfectant as well.
Tropical race 4 disease, it's a very destructive disease of bananas and plantains. It is caused by a fungus, which is soil-borne, and one of the spores, the chlamydospores, it can live in the soil for up to 30 years. So you know that means it's a very deadly, devastating disease. Tropical disease race 4 affects all our local varieties, which includes Valerie, Lacatan, Granane, Robusta, William, and all our local varieties. And plantains, also plantain, horse plantain, horn plantain. These are all the Cavendish varieties. What are the symptoms of this disease? How can you identify tropical disease race 4? First, one of the external symptoms is yellowing of the leaves. Yellowing of the older leaves, right? As you can see here, yellowing of the older leaves, and then it moves towards the younger leaves. See here, yellowing. And also, it doesn't affect the young plantlets or the suckers. Symptoms will be shown in plants that are 4 months or older. So the younger plants can be asymptomatic. That means it does not show the symptoms. So you can see a plant here which is about 4 months old, and you can see the yellowing in the older leaves, and it gradually progresses towards the younger leaves. So that is the difference between tropical disease race 4 and the mucker disease. Mucker disease, the symptoms begin from the younger leaves and move towards the older leaves. Tropical disease race 4, the yellowing starts from the older leaves and move towards the younger leaves. So, and the sucker here, you can see a young sucker, there are no symptoms. It's asymptomatic. It may have the disease, but the symptoms are not yet established, not yet seen. All right, so when the symptoms are shown in the older leaves, after a while, these leaves will die and it will form a skirt around the trunk, the pseudostem or the trunk of the banana plant. So you can see the fall of the older leaves and it forms a skirt around the trunk of the plant. Another external symptom is the splitting of the pseudostem. You can see here the pseudostem are split, and splitting commences from the base of the plant and it moves upwards to the top of the plant. And the pseudostem is the trunk. We all know the pseudostem trunk, fall stem trunk. The internal symptom now, you can see here, it is in the pseudostem. When you cut the pseudostem longitudinally, you can see where the spores, they damage the vascular tissues or the water conducting vessels, the xylem vessel. When this vessel is damaged, that means water can no longer be transmitted up to the trunk of the plant. So that is why you have wilting of the leaves. No water, the leaves will wilt. You can see here the discoloration of the tissues. And it starts maybe with a yellowish discoloration, and when it gets older, it moves brown, dark brown, as you can see here. And this is a cross-section of the pseudostem, or the trunk. You can see where you have the discoloration. And if you notice, the discoloration commences from the outer leaf sheaths. That is why you have wilting that starts from the older leaves moving towards the younger leaves, right? This is also showing the discoloration of the xylem vessel, the tissues, vascular tissues. This is the rhizome or the corm of the banana. This is the section that is closest to the ground, that's within the soil. So this is the corm, and you can also see the discoloration of the vessels here. moved from the outermost part and it moved towards the inner part. Okay. All right. What is Jamaica doing about tropical disease race 4? Some of you may ask. What are we doing about tropical disease race 4 to keep it off our shores and to prepare ourselves for when if it should arrive here, what will we do? First of all, we are strengthening our border security protection at our airports and our seaports, and quarantine is playing a major role in this strengthening in this capacity. And we have prohibited the importation of plants or plantlets of banana and plantain from TR4 disease countries or countries that are suspected of having the disease. We are also trained our scientific officers, our extension officers, our quarantine officers, customs, the Jamaica Constabulary Force, the Fisheries Division, and most of the stakeholders in the banana industry. Yeah, and the Pre-Del Larson Division of the Jamaica Constabulary Force. They have been trained, and we also have awareness sessions across the 14 parishes of Jamaica. We have awareness sessions with our farmers, students, our agronomists, all our scientific officers, and the agricultural industry on a whole. We have awareness session, and also to the general public. And with the assistance of the government of Jamaica, we bought two equipment, state-of-the-art equipment that is used for detection of TR4, rapid in a rapid time and accurate. And accuracy is also involved. And today we are having our first national simulation. This is to really training of our officers, and it is really for us to be able to see our weaknesses, our weak areas, and where we can improve on these areas. So if the disease should get here, we are better prepared to deal with this disease. And I just want to emphasize that tropical disease race 4 is not in Jamaica. This is just a simulation exercise for us to be prepared for the disease. If it gets here, we know exactly what to do. We know how to respond in the various areas. So again, tropical disease race 4 is not in Jamaica. And I will now move to Mr. Lewis, who will tell us more about our site here. As you can see here, we have our infection site, our simulated infection sites, and he will go more into that. Mr. Alfred Everett, he's one of our surveillance officers, and he will be telling us about how the field is surveyed, what you look for, what you do. So I will now move on to Mr. Ora Lewis, who's our biosecurity manager. Mr. Lewis. Any questions?
Okay. All right. Thank you very much, Debbie. All right. So what we have on the chart here, basically a semblance of what they're looking at. And so the key thing here is that we have our infected plant, which of course we refer to this area as Zone A. If you look further there, and designated red as a red zone, right? And then you have Zone B. It's from the infected plant to Zone B. It's actually 20 meters, and then from the edge of, and then we have Zone C, which is 50 meters from the infected plant. In zone, so there is we call a buffer zone around zone A, right? And here you will have designated trenches over here. There's a trench actually around zone A as well. But what is very critical is at this point here. So we're looking from this end, kind of upside down. But at this entry point up to zone C, what is critical here is that there are two baths there. One with water, basically to remove soil material from your feet. And then the second one has the disinfectant, the same disinfectant we spoke about. That is glutaraldehyde and cotton ammonium compound. And then we'll be followed by a dry area, and there we have a sponge. So it is our biosecurity point 1. Then 2, you'll also have at the entrance to zone B, there's another foot bath with disinfectant. And likewise, at the edge of zone A, there's also another foot bath. What is critical when that time comes is that we're going to observe the operation taking place in zone A, but we're going to ask everyone to space themselves around on the edge of zone A. So nobody will actually be in zone A. We're going to be on the edge of zone A, which is in zone B. Okay, is that clear? Yeah. That is at the prescribed point. What will happen shortly is that Sir Everett will demonstrate how we carry out surveillance, right?
And in other parts of the country, in the rest of the world, they use the Greek guard method, where they walk 3 rows at each zone, 3 rows and at a pace where they can observe the plants properly. But in Jamaica here, because we are dealing with smaller farmers, we actually walk each rows. So I'd walk at a pace, I'll observe plants left and right. And if I come across a suspected plant, the first thing I'll do, I'll take my camera out and I'll take the upper section, picture of the upper section, the middle section, and the lower section of that plant. And after doing that, I'll get my spray paint to mark that plant. After marking that plant, I'll also make records on the sheet of the different things that is on it. On the sheet, you have the date, the time, the owner of the farm, the person collecting it, and the parish. And then I'll also use my GPS machine to collect the geo position of the farm to get the longitude and latitude of the location of that plant. And after that, I'll use my spray bottles before I exit that field, sanitize my feet and exit that field.
Good morning again, everybody. I am Sherry Clark, and I'm the Senior Biotechnologist here at the Banana Board. And with me, I have Mr. Henry Graham. He's one of our field officers, and we are trained to collect samples. All right, after the surveillance activity has been conducted, and the suspected plant, which is this plant, has been identified, then I, along with a trained field officer, will be notified and we will visit the farm. But before upon entering the farm, we introduce ourselves to the farmer, put them at ease, tell them that we're the ones to collect the sample, as well as we have a sample collection form with information such as the name of the farmer, the address, the telephone number, and most important, the GPS coordinates, because we want to ensure that when we leave the spot, when the eradication teams come back, they can find the plant and completely destroy it. So we enter the farm, and it is very necessary that we conduct the collection under sterile condition. Because the nature of the disease is soil-borne, what I would do is come in and I have a piece of plastic, and this would be where I work from, so as not to contaminate my sample collection equipment. So I'll spread it around the plant, and then I'll rest my equipment on it. So we have our igloo to transport the sample back to the lab, our kit, and before I start, everything should be sprayed off. So I'll remove my equipment. I have gloves, a garbage bag, duct tape, my forceps, sterile envelopes, the knife, as well as my marker to make any. All right, so I have my sterile paper towel because the sample has to be transported moisture-free, so it is placed in a sterile paper towel. All right, so yes, so we sterilize it in the aluminum for transportation. The sterility will be maintained when it's being transported. All right, so we go ahead, and Mr. Graham will now demonstrate. What we do is, from the base of the plant, just a little bit above, we cut a window. This window is about 3 centimeters deep, and it's 5 by 10 centimeters. And what it does, given the nature of the disease and how it spreads from the outer vascular towards the inside. So this is the method of choice. So remove the window, place it on the bag. And to note, this is not an infected plant. It's a fairly healthy plant. But what we look for when we're in the field is that the vascular strands are supposed to be discolored. So when we remove it, we'll see reddish brown or dark brown. Hold it. So what I'll do for transportation purposes, remove the excess pseudostem or trunk, and I'll transport only the vascular tissue. So this part, that's the part I want to take into the lab. Pass that. Care has to be taken, so it's not a case where a rush. You have to take your time. Are you correct? And what I do is remove the sun hot. I'm kind of overwhelmed a bit. Okay, so what I'll do is peel off the pseudostem. And this part is the part that are taken back to the lab, the vascular strand. So, yes, so I remove the strands. So this is a vascular strand. So you would see a reddish brown, you would see the reddish brown or dark brown. The strands would look discolored. So that is what we want. Show them.
This is what it looks like. So scientifically, you don't mind.
All right, so this is a vascular strand. All right, so it's supposed to be, for overseas, they don't want any moisture in it. So they use the liquid nitrogen and they would grind out the cells and then extract the DNA. So that's the protocol we've adopted. So place it in a paper towel, put it in an envelope, put it in the, so I'll show you. Paper towel, envelope. And then, because it's going in the ice, you don't want it to get wet, ziplock it. And it's labeled, you see sample A, Borders Old Harbor, and the date. So what we have is a conventional PCR, and that it looks for the target. So when we amplify the DNA of the sample, we compare it to that standard. And if it's present, then absent or minus. If it's present, then we have the disease. If the TR4 gene is absent, then we don't have the disease. The shortest one we have is the LAMP. And this stands for loop mediated isothermal amplification. And this can be done in 6 hours because it's a 1-2 process where the reagents are placed in the tube and it seeks in layman terms the TR4 gene. If it's not present within the sample, in real time, it won't be amplified. If it's present, then we'll see a peak, and that will indicate that the disease is present. The real time, however, the real time and the conventional will take 2 to 3 days because you have a pre-run and a post-run as well as a quantification. So what we use for fast diagnosis is the LAMP because it is 6 hours. So that will give us an indication. All right, we need to get on the sample. If we find a positive sample within the lab, it is then sent overseas to our confirmatory lab, and they will then let us know that yes, it is here. The DNA sample itself is sent, and they will do their testing. We do not wait on the confirmatory test, according to just what Mrs. Coney said. If we get this 2 to 3 tests are positive, then the team will come in, and they will completely destroy the plant. So we don't wait. So as soon as we get the results in the lab, if it's positive, we communicate that, as well as communication to the farmer is necessary. So the farmer is aware that yes, you have it. If not, good, then that is good. But we communicate that information to the farmer as well as the eradication team. All right, so after this is finished, we have a garbage bag, and all our utensils are placed in there. No, left those, leave those. So these. No more. take that one. Alright. Alright, so the plastic as well as the excess sample, they are left in the field. We don't take this back. So as to prevent contamination, because we'll be going to, hopefully, if we doing the surveillance, we'll go to other farms and we won't reuse this plastic because we're not sure. Alright, so say sadly we get confirmed the disease in the lab. Then the eradication team will come in. So we have Mr. Yates and Mr. Clive, and they work here at the Borders Research Station. And what they would do is the now infected plant, but it's no longer a suspect. We're partially like 90% sure that it has the disease. So what we go in and they will completely destroy this plant. So what they'll do is dig it up, cut it up, and put it in a black bag. Black bag. After this is done, then they'll add 1 kg, which is approximately 2.2 pounds of white lime and urea to this bag. After they get rid of the infected plant, then the other plants in zone A, which some may not display symptoms, they then dig up those, chop them up, and they're placed on the ground of zone A. On the ground, then application of 1 kg per square meter of urea and white lime is placed on the plants, and then a plastic is used to cover the area, the entire zone A. After this is done, then a trench is being cut along the borders of zone A, and this is to prevent runoff. So you don't want the water running off from zone A to contaminate the rest of the field. So the plastic is placed, trench is dug, and then the edges of the plastic is being covered with the dirt. So that you'll see that being demonstrated now.